Hello friends, welcome to BMH Learning. This video is about sequence assembly, also called genome assembly. Genome assembly refers to the process of putting nucleotide sequence into the correct order. In bioinformatics, sequence assembly refers to aligning and merging fragments from a longer DNA sequence in order to reconstruct the original DNA sequence. This is an essential step of the genome analysis process because the entire genome cannot be interpreted in one step with current sequencing technology. Instead, small sections of the genome with up to 30,000 nucleotide bases are read at once and then assembled to reform the DNA. Assembly is a difficult task because there are often many different fragments of the genome which need to be pieced back together in the correct order to make sense of the information. There is also some room for error in the assembly process due to the repetition of the nucleotide bases in the genome, which may increase the difficulty of assembling the fragments together correctly. The alignment of multiple fragments involve overlapping the ends of the fragments. Approaches. There are two broad types of assembly techniques that may be utilized: de novo and comparative assembly. De novo assembly. It is used for new genomes that have not been previously sequenced or are not similar to genomes that have been previously sequenced. This type of assembly is usually harder to conduct due to computational difficulties. Second is comparative assembly. It is used for genomes that have an existing sequence or have a genome that is similar to another organism that already has an assembled genome, which can be used as a reference. This was all. Thanks for watching.